Hello, welcome. Uh, this movie will walk you through some of the details of ultra small angle scattering instrument and the data provided to users and how to get them easily into Igor or any other applications. This movie is really useful for either current, past, or future USAXS users. And unless you want to use my instrument, um, probably don't need to bother about watching it. So first thing is, what is a USAXS instrument? The ultra small angle scattering instrument at the advanced photon source is an instrument which I've been maintaining for about 25 years now. It's a unique instrument which provides a wide range of data which you can measure. This is the web page, usaxs.xray.apsanl.gov, and here is the web page which describes what it is, how it does. The main thing is that we provide a very wide range of sizes which can be measured from about 10 to minus 4 inverse angstroms to 6 inverse angstroms, which in sizes goes from few microns to about 1 angstrom in a real sizes. We have also high resolution mode, which goes even over broader size. This is achieved by combination of 3 different devices. The main device is the USAXS, the ultra small angle scattering instrument, which is basically a high resolution diffractometer measuring at low Q or around Q equals 0, the main direct beam. We also have a USAXS chamber, which measures higher angles, and then we have a powder diffraction device, or we call it a WAXS, wide angle X-ray scattering, and that measures even wider angles. And if you look on what we do is we measure sequentially 3 different areas in angles, producing 3 different curves. The red one in this picture is the USAXS, and you can see it starts about 10 to minus 4 inverse angstroms and then goes to about 0.2, 0.3 inverse angstroms. So typically we measure about 3.5 decades in Q, 1, 2, 3, 3.5 decades in Q. In a high resolution mode, we can actually measure 4. Then we have a USAXS instrument, which typically measures from about 0.03 to 0.05 at low Q and goes to about 1.1 inverse angstrom. So it covers the highest range of USAXS region normally. And then we have the powder diffraction device, which gives you the standard peaks up to about d-spacing of 1 or less than 1 angstrom. So you get 3 different measurements during our data collection. And so with these 3 different measurements, you actually do generate multiple data sets. And this is where some of the issues with the with our instrument data are. Okay. Now I'd like to point you to some of the other resources. Somewhere on this page you can find a link to a very useful manual page. The manual page is here, to manual page, and in here all of the things which I will be describing are described in some of the first pages here. So you get instructions about what is the instrument, something about the history, data collection, and then it goes through folder structure, metadata, how is the data being reduced, about Matilda, which I need to talk about today, and so on. So all of this information is also in a written form in the Indra and Nika manuals, which you can find on Reader Docs, and you can find the links everywhere. Most of these links you will find are on the USAXS webpage in a software area. In this case, it will be Indra, which is the data reduction package for the USAXS instrument itself. Notice that currently, there is a January 2026 beta release of the tools, and what I'll be talking about is actually in these tools in the beta release. So if you want to use this thing here, you're gonna have to install the beta version there. Now, when you collect data on the USAXS instrument, there are instructions how to schedule the data collection. Basically, you have to collect an instrumental curve first, and then you have to collect the data. And for most users, not all, but for most users, our automatic data reduction routine, which we have available, will properly reduce and calibrate and desmear and process the data on its own. So at the end of the measurement, within 15-20 seconds after the measurement is done, we have a fully calibrated available data. Um, the data are collected for any user. We will create a folder which will have a date. For example, this is November 23, and then it will have a typically username. Um, that's a folder which contains everything measured for that given user at that time. And if it's a mail-in program, there may be a few measurements. If it's a large program of a week of measurements, there may be lots and lots of data. Inside will be some text files, potentially some Igor experiments, but then the user does have a control over the internal structure. So there will be an internal structure of folders. So in this case, the default are data. So there will be a folder data, which we start with. And then anytime the user changes a sample to something meaningfully larger, which contains multiple measurements, they can create their own separate folders. So in this case, we had a user who was collecting something which was cast MAR 247 and so on. Now I said we have three different segments of our instrumentation which measures sequentially, and so we also get you three different data sets of each sample. The way that's structured is inside a data folder or inside any other folder will be a folder of the same name, data, underbar, and is going to have a USAXS, USAXS, and USAXS. Inside these will be the measurements for those little segments. And so inside, for example, the USAXS, you will find that there are files, and typically there are 2 files per sample. And that would be the HDF5 file, or with the various extensions, H5 or HDF, depending on which segment it is. There's a reason for that. And then typically we also collect an image of the sample before the measurement. So before each measurement, we snap a picture. And that helps us to provide a trust. This is what was in the measurement, in the instrument before your measurement. And so in this case, you can see that you have an air blank 12 kiloelectrons and 21 kiloelectron volts. You have a glassy carbon standard reference material measurement and so on. If I open the USAXS, you will find out that there are similar or same measurements. Typically, we also have a calibrant in the first folder here. So that will be the silver behanate lab 6 measurement. And then we have the same thing in a USAXS. So if you look in a USAXS, it's here. And so each one of the sets of measurements does have all of these 3 segments at the same time included in there. So when we share with you, typically we use a box. That's the easiest one because our sample measurements are typically relatively small. No one produces terabytes of data out of the USAXS instrument. So if we are looking on a few hundred megabytes or a gigabyte of data, a network storage like cloud provider box, which Argon makes available for us, is actually easiest to share. So if we share with you this folder here, inside you will find all your data, any Igor experiments you were using during the data collection, and this .dot file is the record on that. Now, what are these HDF5 files I keep talking about? HDF5 files are files which there's a whole website on that. This is a data storage system for high performance computing and for scientific data. So if you want to read about that, the HDF group will give you plenty of information about that. Suffice to say, it's a binary container which contains the data, but it's a container with very rich structure inside which you can have a complicated structure, kind of like folder structure with various components like strings and arrays and multidimensional arrays and all of that stuff. And so it is very convenient because you can take and put a lot of different things which you can properly identify, give them units, give them all sorts of metadata, and then provide them to user as a one big file. Our data follow 2 different standards for small angle scattering. So there's an NXSAS standard. This is a group of scientists got together and created something called Nexus format. Nexus format is basically an agreement on how are we going to structure the data inside and what type of metadata and keywords are we going to use and how are we going to attach things. So different applications can understand the data. And so for raw small angle scattering data is a standard NXS, which our SAXS and USAXS are both using. So we record the metadata and the area detector image and everything else based on this standard. There's no standard for USAXS, so we created something very similar but suitable for the Bonds Hart instrument we have. We also have an NXCANSAS, which is a definition for reduced data. For small angle scattering, you can attach to the same file, you can attach raw data and reduced data, and applications can understand it. So if you have an application which is supposed to reduce the raw data, it can understand and pick up from that file the raw data, whereas if you have an application which looks for reduced data, it can pick the reduced data. It can actually document. It's kind of cool. So I showed you that we have the data here, and these are USAXS data, and so I told you this would be a data set which has a raw data and now also reduced data using our automatic data reduction. So we have an application. In this case, this is a SAS view. SAS view can read and understand the NXCANSAS data. So if I take this file and I throw it in SAS view, you can see that the SAS view understood, it imported that, and I can send it, and I can send it to a plot, and it will plot the data. So without any data reduction at all, your USAXS is already reduced. That has been done by the automatic data reduction routine. And so if the standard parameters are fine for what you need to do, for what you want from the instrument, and if you don't need to do any changes, the data directly provided without any further data reduction contain calibrated, desmeared, and corrected, and everything. They're just ready to go data inside each file, which are written according to the standard. So a standard adhering consumer application like IRENA in Igor, or like SAS view, or anyone else, or Python program, you can read them via Python interface, can pick up the data and just use them directly. There is no further data reduction needed at all. That's not completely true if you have needs to change the default parameters, or if you have specific needs for different type of calibrations, or if you did not follow the proper instructions on the proper sequence of data collection, that you need to collect the appropriate blank first and as a last before all the measurements of the samples, appropriate ones, then you may need to go and redo it in Igor if you didn't do something right, or if you need something else. But let's go further out with this. So what was the next thing here? Okay, so just few notes on this. I'll get moving on what to do next. But there's a GitHub page which you may want to know about. This is where the IRENA, Indra, and Nika are. And if you find SAS Igor code in my depository, you can find the current versions. So the current version, stable version, is July 25, but I have released this January 26. The beta version. Similarly, you will find that same instruction here on the USAXS homepage for software, where you find the current release, which is here, and the beta release, which is here. Now, this new release is really useful mainly for users of the instrument from the last cycle. They do have the reduced data. They can take benefit of that, and they can take advantage of what I'm going to show you next. So let's put that away and let's go to Igor. So most of users will be using Igor, presumably. So I have an Igor Pro, in this case an Igor Pro 10. The current beta release will support only Igor 9 and Igor 10. Igor 9 is the last version which seems to be available for Mac. They don't have, Wavemetrics doesn't have plans to support Mac on higher versions in Igor. So I am actually running a virtual machine, virtual Windows machine, in which I have installed Igor 10. I have also, and it's the beta version. So if you now want to load the USAXS macros, you find in macros, you go in and say USAXS. And if you need to install it, the instructions are in a different movie. So you just find the movie here in this YouTube channel and follow the movie there. So now when you start the USAXS data processing, it now opens Indra, Irina, and Nika all together. So you have the USAXS, you have the SAS 2D, which is Nika, and you have the SAS and BioSAXS, which are the Irina. And the reason for that is we just need them all. Okay. So the new tool which I want to show you is the USAXS SAXS WAXS data reduction new. I need to find something shorter in there, but anyway, that's the new tool which I'm going to be showing. The old one, for which there's yet another movie in the YouTube channel, is completely valid, completely functional. You can use it. If you know how to use it, you're welcome to use it yourself. That's perfectly fine. That's available here under the original, okay? But let's go to the new one. The new one panel for now this is a beta version, okay? I am not yet adding some of the other functionality in there, even though it might partially work. Don't try to use anything beyond what I'll show you, which is basically import, okay? And the reason for that is that I just don't have time to do it fast enough. Anyway, so here is a interface. We're gonna point ourselves to a folder, and it should be on a desktop, and that's the 1123 username. Let's go after any one of these. And now you can actually point yourself to any one of these, SAXS, USAXS, or WAXS, it doesn't matter. And in some way it matters only if you did not collect for every USAXS also SAXS and WAXS. You want to point yourself to a folder where you have most of the measurements, and you're going to understand later why, but basically, in some cases we have users who want to collect, for example, during heating cycle at the beginning only the diffraction pattern, only the wax, because you can do it basically every second or every 2 seconds. So they get a very detailed understanding on how the phases change, and then when they reach some temperature they want to put in USAXS, wax, and so on. If you point yourself to a folder which does not contain anything about those wax measurements, about some measurements, you will never import them. But if you point yourself to the folder with most data, you will always import all of those and any existing from the other folders. So in this case, we're going to go to USAXS, select the folder. We now have all of these in there. So now, and you get the standard beautiful things here that you can go in and you can do if you have complicated name system, you can actually use a match string here with a grep string here, and you can mask off some of these. Next thing you need to decide is what do you want to import, because remember, you have USAXS, you have SAXS and WAXS. You may not care about some of those. And also, by the way, you may or may not have the images, okay? So in this case, let's just pick, I want to import USAXS, SAXS, WAXS, and an image. Note this re-reduce data, that is not functional yet, so wait until the final release, probably May or June. Okay, and let me select any one of these and say import. And when you hit on this import, all it does, it goes and grabs every one of those HDF5 files, and it will go in and it will import the data. And so it will import the USAXS data, and if someone remembers how the data are reduced, this is a step in the data reduction. So these data are all there, all imported. They're not important for you, but they're all there now. So this is an instrumental curve and your sample measurement as a normalized but not subtracted data. Then the instrument produces what is called slit smear data. So it normalizes, it subtracts those two data sets and applies a calibration constant. So these are data on absolute intensity scale. You can see it, it has unit centimeter square per centimeter cube, which is the standard unit. But SMR means it's a slit smear data. You could use in IRENA slit smear data perfectly fine. IRENA knows how to slit smear the data, the model, and then fit to slit smear data. So this is already usable data which you can use in IRENA. By the way, you can also use them in SAS view, which knows how to handle slit smear data, and a standard does have a method how to describe the data. So the slit smear data can be used directly already by SAS view also as a slit smear data, and it will properly slit smear the model. But then the last pass of the Matilda data reduction is it will run through a desmearing routine, and so this produces a USAXS calibrated desmear data. This is the pinhole equivalent. If you want to use any other tool, or if you want to use your Python code to do some modeling, analysis, anything else, this is the data which you want. They're already in Igor. They were just imported in. There was no calculations done. It was just a simple import. Then we have a SAXS data. This is the SAXS data segment. So you can see it goes from 0.04 to about 2 inverse angstroms. This is probably higher energy. And so this gives you the high angle, small angle scattering. And then this is a powder diffraction thing in Q units. So this overlaps with the SAXS in this area. So you can stitch that together and goes well over 6 inverse angstroms. So that gets you to 0.8 or so in D-spacing or something like that. So now you have all of those in there. And by the way, it shows you the image. In this case, it was a sample inside the Lincoln heater. So you can go in and say, well, I want to get more of those. And you can say import. It takes very little time, and it puts all the data inside Igor. So if the approximations or decisions on a number of points, calibration method, and a few other things are appropriate for your data reduction and calibration, which mostly probably are, then you don't need to do anything. The data are already available, and you can go ahead with this. You can also just import some of them. Let's say that you don't care anything else than about Igor. You can go about USAXS. You can just go in and import them all as a USAXS. So that's convenient. Just to show you they are there, I'm just going to go in and say, why don't we do a plotting? You go to USAXS. We pick on a USAXS, and you can see these are your USAXS data. Select and create a graph. Here they are. You can also select the slit smear data. So you can do the same thing, but this time with the slit smear data. So you're looking on different slopes and different intensities, but that's because of the slit smearing involved. So everything is in there. Now, as a final step, if you just want to import a whole sample, what do you do? Because this means that you have to go through all of these, and now you may actually not pick the right folder, and you may be missing something. So there's another basically wrapper around all of these functions. You go in here, say import whole sample. This time, if you want to do that, you don't point on a segment, but you select the folder which is above. So you now select the folder which contains the 3 additional folders. You select the folder, and the code just going to go in and look inside each one of those subfolders and import all of the data. That may take some time because it's reading a lot of data, and then it replots them immediately. And now you have all the data at once. So if everything works well enough, all you need to do is go and open the folder with the subfolders, import the whole sample, and everything should go in. Now, the data and this will give you, all of these beautiful graphs. One thing which this does not do, and for now we're not even trying that, is merging the data. And there's a movie on how to do that, but remember, now you have the 3 segments. So if you look and say, okay, I want to do USAXS, desmeared, so that's a USAXS. Then you can take on the first one and you can do the SAXS, That's your USAXS, and then we can do the USAXS. Okay, so now we have, we have the red, blue, and green, or whatever is the same as is on the website. And you can see that there are some, there's a background in here, so this needs to merge together somehow. It's a lot of processing to now merge what oftentimes people want to merge, which is the USAXS and USAXS. There's a tool in Arena for that. There's a movie for that. You can go through it and you can do it, but you have to do it separately. That does not look like it's doable really easily or reliably in other tools, in automatic tools. Okay, so by now I think I did all of I wanted to tell you here. So we now have a new tool, Indra, for import reduce data package. It will eventually be able to reduce the data too, so you can, and actually already can do it, but I'm just not sure if it does it robustly and reliably. I'll be testing that. And it will eventually be able to change some of the parameters. Currently, when you take the USAXS data, we collect 8,000 points in USAXS, but that's too much for any reasonable calculation. So we, by default, bin it down to 500 points. That's mostly okay for practically anyone, except for people who have monodispersed systems or something with diffraction peaks or, more complicated structures, in which case the number of points can be rebinned manually or can be changed in Igor later on, because you can go all the way up to 8,000 point or 7,000 point or so. But that has not been implemented as an option in the automatic routine. The automatic routine has to work without any user input. There are a few other things which needs to be changed or may need to be changed for various of these steps. So the reduce data checkbox here, while it's active, I strongly discourage anyone from using it. And that's about it. So that's a new tool. It makes life of users of the USAXS instrument significantly easier. And you may not even need that if all you need is a USAXS data and are using either your own Python routine or using SAS View or anyone else who understands the NXCan SAS standard in the HDF5 files, which could be by now 3 or 4, maybe 5 different applications. Anyway, so that's it. Thank you very much for attention. Keep in mind that if you want to apply for USAXS Beam Time, you want to first talk to me, let me know, ask for input. We have tool, for example, which can calculate scattering from a model. So I can help you pre-calculate how your scattering should look like, if it's measurable, if we can, see something. I can help you decide if that application or an application for beam time is meaningful. So just get in touch with me if you want to measure your data. If you don't understand something, get in touch with me too. And by the way, this is a beta version, so if you run into any troubles, send me as much description and information, and ideally also Igor experiment, so I can figure out what's wrong and debug this for the future. And that's it. Thank you very much. See you future.